Welcome back to Acing AP Biology. In this lesson, we will discuss the process of translation, converting mRNA into a polypeptide. In translation, the mRNA transcribed from DNA is translated to an amino acid sequence, which is the primary structure of a protein. The mRNA attaches to a ribosome where the translation process begins. mRNA nucleotides are divided into codons, which are triplet sets of nucleotides. Each codon codes for one amino acid. The stages of translation are initiation, elongation, and termination, just like in transcription. Transfer RNA also have an important role in translation. tRNA acts as a mediator between the mRNA codons and amino acids. The tRNA links the correct amino acid to the specific mRNA codon. tRNA molecules are specific for the amino acid. One end of the tRNA molecule is bound to the specific amino acid, and the other is attached to the anticodon that correlates with the mRNA codon pairing with the amino acid. Think of tRNA as the molecule that interprets the mRNA code to form the specific polypeptide. tRNA is composed of a single-stranded RNA that is folded into a clover leaf shape. Hydrogen bonding forms between base pairs and double-stranded regions. The amino acid binds here at the 3' end. There are about 45 different types of tRNA molecules. Some tRNA molecules pair with 2 or 3 mRNA codons. Now that we've discussed the structure and function of tRNAs, we will go over aminoacyl-tRNA synthetases, which is an enzyme that catalyzes the linkage of tRNA to its specific amino acid. Each amino acid has its specific aminoacyl-tRNA synthetase. This process requires ATP because it is endergonic. First, the amino acid binds to the enzyme's active site, and ATP binds. ATP loses 2 phosphates, becoming AMP. The matching tRNA then binds to the amino acid, replacing AMP in the enzyme's active site. The tRNA is then released, bound to its proper amino acid. Ribosomes are essential for translation. Ribosomes are protein manufacturing organelles. They mediate the tRNA anticodon pairing with the mRNA codon. Ribosomes are composed of a large and small subunit. They have 3 different binding sites for tRNA molecules, P, A, and E. The P site holds the tRNA molecule attached to the growing polypeptide chain. The A site holds the tRNA with the next amino acid to be attached to the growing chain. The E site is where the tRNA, no longer bound to the amino acid, exits the ribosome. Let's now discuss the first stage of translation, initiation. First, the mRNA binds to the small ribosomal subunit at the mRNA binding site. The initiator, tRNA, carrying the starting anticodon UAC for the start codon AUG, binds. Once this takes place, the large subunit of the ribosome binds to the small subunit, and the initiator tRNA fits into the P site, and the A site is now available for the next tRNA molecule. GTP provides the energy for the ribosome assembly. Initiation factors that are proteins also aid in assembly. Next comes elongation. Elongation can be broken up into 3 phases: codon recognition, peptide bond formation, and translocation. Firstly, in codon recognition, the tRNA molecule holding the complementary anticodon bind to the appropriate mRNA codon in the A site on the ribosome. Only the matching tRNA molecule can bind to the codon. The P site holds the tRNA molecule with the growing polypeptide chain. The hydrolysis of GTP into GDP and an inorganic phosphate enhances the efficiency of codon recognition. The next phase is peptide bond formation. GTP is required for this step as well. The new amino acid in the A site forms a peptide bond with the carboxyl group of the growing polypeptide strand in the P site. This process is catalyzed by rRNA. Now the growing strand is held by the tRNA in the A site. Lastly, translocation takes place. And this process once again requires GTP. As the ribosome shifts to the right to initiate the translation of the next mRNA codon, the tRNA that was initially in the A site holding the growing polypeptide chain is now moved to the P site. And the free tRNA molecule that was initially in the P site is now moved to the E site and is then released. As the ribosome translocates, the mRNA is moved as well. And now the next mRNA codon for the growing polypeptide chain is available in the A site. Notice that the polypeptide strand grows in the direction of 5' to 3', corresponding with the mRNA. And lastly, we have termination. The polypeptide strand keeps growing until the mRNA stop codon UAG, UGA, or UAA is in the A site. Instead of tRNA binding, a release factor binds, as the stop sequence codes for a release factor. The release factor spurs the hydrolysis of the bond between the tRNA and the last amino acid on the polypeptide chain, so both the polypeptide chain and tRNA are released. 2 GTP molecules are hydrolyzed, providing energy for the dissociation of the translation complex. The ribosome separates into the small and large subunits. The release factor and mRNA strand are also liberated. That's it for translation. In the next video, we will discuss regulation of gene expression. See you then. For more bio content, hit subscribe.